bound, they're in one structural state, usually inactive. When they have GTP bound, they're in a different structural state, and what that means is most of the time they're active and then they go off to regulate, bind, and regulate other proteins. So in many, but not all cases, G proteins are active only when GTP is bound, and they're dead in the water when GDP is bound. That's not always the case, but it frequently is. That's another example we got. Now, a third example. Okay, what we call the protein kinase A or protein kinase G family. In other words, we mentioned this before, it's PKAs plus PKGs. We're going to look at these guys more when we talk about cell signaling. And these are important regulatory proteins that actually do covalent modification of target protein. I haven't gotten to that yet, but we will. These proteins are inactive unless they bind a cyclic nucleotide. That's the nucleotide where the phosphate is twisted around a circle and reattached to the carbon above the carbon that it comes off. Those cyclic nucleotides bind and activate these PKA and PKG enzymes. And once that happens, they're active, they go and regulate a whole slew of other proteins by covalent modification. So what we see here, of course, this is only a couple examples, but what we see here is a whole slew of diverse controls and regulation of proteins by small molecules. I'm just going to take this. I gotta keep the file size down. Wait a minute. Okay, good. All right. Now we're going to take a look at one other aspect of regulation by small molecules. And this is important in controlling various kinds of metabolic pathways. And we call this feedback inhibition. Now, what is feedback inhibition? Feedback inhibition is this. You have an enzyme. It binds a substrate or substrates. It does something to it, catalyzes some kind of reaction, and then it produces a final product or products. In the case of feedback inhibition, the product of that enzyme will bind to another spot, not the active site, but to another spot on the enzyme, warp its tertiary structure, and warp the active site and shut that enzyme down. So in other words, the enzyme is inhibited by the very product that it makes. So the enzyme is inhibited by the very product that it happens to make. Now a little diagram of this. We have the enzyme here. I'll draw an active site over here. A substrate binds to it. It does something to that substrate, producing a product. And then that product can go back and bind to the enzyme in another location. Well, let's stick it right here. And it alters the active site, in this case closing the mouth here, and the enzyme shut off. Now, of course, since this is a non-covalent binding, sooner or later the enzyme is going to shake the product off, bye-bye, and it goes back to the active state. Now you might ask yourself a question, why would you want an enzyme to be shut off by the very same thing that it produces?
Make too much.
Excuse me?
So it won't make too much.
Exactly. Dead on, beautiful. Okay, all right. Let's do a little example here. Where do we find this? Where do we find feedback inhibition? It's usually used when you have a kind of metabolic pathway that has multiple steps in it. And many of these things exist. For instance, the process of glycolysis that leads to the splitting of the sugar glucose and generation of little ATP. That takes about 7 or 8 steps. Krebs cycle, also important metabolism. That takes about 12 or so steps. Now, why do you use things like that? Well, let's get kind of an analogy here. Let's picture this as a molecular assembly line. So we go out to Honda or Mercedes or Kia or whatever and look at the assembly line. Here's an assembly line. Okay, we're going to have enzyme 1, substrate 1. He goes out and produces product 1. Okay, now product 1 is the substrate of enzyme 2. And it makes product 2. And then product 2 is now the substrate of enzyme 3. And it makes product 3, and that becomes the substrate of enzyme 4, and it makes product 4. Okay, so we have a molecular assembly line. Now picture on an assembly line, in fact, you're going to have some people that are really active go-getter types, right? And then you're going to have some slacker types. If you have an assembly line, and let's suppose enzyme 1 is a real hard-driving go-getter type, and he's going to bust his butt for 8 hours a day and hopefully get decent paycheck and benefits. It's rare down here nowadays. Okay, but enzyme 2 is a slacker. Hey man, you get what you pay for. You exploit me, I'm going to get back at you. Okay, so what happens? Enzyme 1 is inherently faster than enzyme 2. So enzyme 1 is working a lot better. These different enzymes catalyze the reactions at different rates. Some are very fast, some are very slow. So, if enzyme 1 is fast and enzyme 2 is slow, what's going to happen? Enzyme 1 is going to start producing more product 1 than enzyme 2 can handle. So what's going to happen? Product 1 is going to build up faster than enzyme 2 can get rid of it. Now there's a downside to that. One thing is maybe this product, obviously you're wasting substrate and probably energy producing a whole bunch of product that enzyme 2 can't keep up with. Second thing, sometimes these intermediate products can actually be toxic if they build up too much. So now you got a situation here. It's nasty. You're flooded with product 1. That enzyme 2 is just taking its good old time here. Yep, hey, I'm going to have a good lunch break for 2 hours and sing right afterwards and stuff. Whatever. Okay, well, you got a problem here. Now, unlike an assembly line, they can't fire enzyme 1. So what we have to do, this is where feedback inhibition comes into play. Because now, let's suppose enzyme 1 is regulated by feedback inhibition. Our product binds to it, and we temporarily shut the enzyme off. So, this enzyme is inhibited. Meanwhile, enzyme 2, the slow guy, can start catching up on the backlog. So now we have less product 1, enzyme 2 is trying to catch up. Now, the nice thing about feedback inhibition, because this is non-covalent binding, is this. Each time product 1 binds to enzyme 1 and shuts it down, it's not going to stay there forever. It's going to get shaken off. So bye-bye. And now enzyme 1's turned back on again. But if there's still a whole ton of product around, it's very likely that a second product 1 is going to bind and shut the enzyme down again. If there's a whole bunch of product 1 around, it means most of the time that enzyme is going to be binding to it and shut off. But on the other hand, when the level of product 1 drops down to something a little bit more reasonable here, you shake the product 1 off, and there's very few product 1s left to bind to it, so the enzyme is on most of the time. And then once you start getting a little bit of a pile up and stuff, then we start shutting the enzyme off. So with feedback inhibition, it effectively acts as a concentration sensor. If you have a lot of product 1, enzyme 1 is going to be shut off most of the time. If you have a small amount of product 1, enzyme 1 is going to be on most of the time. So the purpose is to prevent unwanted buildup It's going to prevent unwanted buildup of a particular product. And that allows, by having in these multi-step enzyme pathways, regulating at least a couple of the fast enzymes by feedback inhibition, it means you're going to coordinate the activity of the whole thing. So you don't have buildups of any kind of intermediary products. You just have the thing, whole system, the whole assembly line running nice and smoothly. And that's the cool thing about feedback inhibition. It allows you to coordinate the activities of both fast and slow enzymes so that nobody gets ahead of anybody else. And you keep the whole pathway running nice and smooth. So glycolysis is regulated at points by feedback inhibition, so is Krebs cycle, and so are numerous other types of metabolic pathways. So that is an example of regulation by binding of small molecules. In this case, the product of an enzyme binding and inhibiting the very enzyme that made it. Okay, so that's another type of thing. Okay, so I'm going to switch again.